Today's lecture is going to be involving the idea that we've already learned about actin and myosin, and troponin and tropomyosin. We've learned about myofibrils and the special things that go on inside of the muscle fiber. We're ready to move on to muscle physiology. And the better you've understood that first set of things, the microanatomy of the muscle, the better you're going to be able to understand the microphysiology of muscle contractions. So the question today is, what causes the proteins, these actin and myosin myofilaments, to begin moving to overlap each other? And it turns out two external things are needed, calcium and energy. We're going to start with the calcium story. A muscle fiber has a specialized sarcoplasmic reticulum, which you can see in blue in this picture. We're looking at the inside of a muscle fiber where we've pulled out 5 myofibrils, these rod-like protein structures. You can see that this blue netting here is wrapped around the entirety of each muscle fiber. I'm sorry, each myofibril, I apologize. Each myofibril has these sarcoplasmic reticula wrapped around the entire thing. And even though a small part of this is shown, it actually runs the entire length of the myofibril, which in turn runs the entire length of the muscle fiber at once. The sarcoplasmic reticulum is the approximate equivalent in other cells of the smooth endoplasmic reticulum, which is mostly in charge of storing things. In this case, in the case of muscle fibers, the sarcoplasmic reticulum is in charge of storing calcium almost exclusively. So the sarcoplasmic reticulum runs along the length of the myofibril. It contains a ready-to-release supply of calcium. And this structure here, this little white structure along here, is a T-tubule, standing for transverse tubule because it runs transverse to the length of the cell. And this T-tubule runs along a Z-disc, in each case, and closely contacts the sarcoplasmic reticulum. The T-tubule will quickly transmit an action potential, causing the sarcoplasmic reticulum to release calcium. And the release is incredibly fast, so all of the calcium along the entire length of the muscle fiber is basically released simultaneously. And if you'll notice, the reason this happens is that the sarcolemma carries the charge. It carries the action potential down the entire length of the muscle fiber. And what you'll see here, if you look closely, is that the T-tubule is just an invagination of that sarcolemma. So when the sarcolemma carries the charge, it also carries the charge directly into the T-tubule, which is basically a continuation of the sarcolemma. Now we're not covering this specifically, but I did want to give you the whole story just to complete the picture. So what we're going to start to talk about in a moment is excitation-contraction coupling, the details of this release of calcium due to an action potential. But first of all, I just wanted to show you, this is what we're looking at here. In the top part is a presynaptic neuron. This is specifically a motor neuron that is carrying acetylcholine in its membranous vesicles, and it experiences an action potential, and as it experiences an action potential, calcium influxes through those voltage-gated calcium channels. When the calcium influxes, it interacts with these membranes, which carry acetylcholine or acetylcholine. These membranes then fuse with the very end of the axon terminal and dump their acetylcholine contents into the synaptic cleft. Now, if you haven't noticed it already, this motor neuron is synapsing with a muscle fiber, a muscle cell, usually right kind of smack in the middle of the muscle fiber, and it's going to be dumping this acetylcholine in the muscle fiber itself. The sarcolemma has these receptors that are receptors for acetylcholine in humans or in vertebrates. It has these receptors for acetylcholine, and it basically works as a typical ionotropic or directly working channel. In other words, the acetylcholine binds to the channel pops open, and sodium rushes into the cell, causing an action potential. And you'll see here that it's going to move down the sarcolemma. The reason it moves down the sarcolemma, of course, is voltage-gated channels. You can see your voltage-gated sodium channel here with its inactivation gate, and your voltage-gated potassium channel. And so the action potential works the same way it works on an axon. So it has these voltage-gated channels that carry the charge down the length of the sarcolemma, much the same way an axon does, right? All right, so that's kind of the first step of what happens during excitation-contraction coupling, is that there has to be an action potential. And the big picture here is that an action potential coming from a motor neuron is going to cause the release of calcium from internal stores, in this case the sarcoplasmic reticulum. And there's a bunch of stuff that happens in between there, but the basic idea is that you have to have an action potential before the calcium can be released. And the reason you have to have an action potential for a muscle to contract, or you have to have some kind of an electrical charge for a muscle to contract, the reason for that is that you have to be able to release the calcium from those internal stores. There's really no other way to do it than to somehow charge up the sarcolemma. So the handiest way to do that inside of the body, of course, is to just create an action potential. Okay, so that's kind of step 0 of excitation contraction coupling, or EC coupling. So let's talk about the events of EC coupling. There are going to be 5 that I'm going to be discussing. Okay, the first one is this action potential will make its way down the sarcolemma and into the T-tubules. And keep in mind that that charge has already come from a motor neuron. But now what we're doing is we're discussing just the action potential that has been moving down the sarcolemma, and as you can see here, it moves down the sarcolemma and also dives into those T-tubules. There's only one shown here, but there are going to be myriad T-tubules along the length of the Z-discs, all down the length of the muscle fiber. So this action potential moves down from the neuron down to the T-tubules. Now, the second step is that the sarcoplasmic reticulum releases a flood of calcium into the cell, and this in turn will contact the myofilaments, which are parts of the myofibril, right? And the calcium then, the step 3, is that the calcium binds to troponin. The troponin will change shape to unbind tropomyosin from the actin. Step 4 is that once the actin has been released from its tropomyosin blockade, its active sites are open. So step 4 is that myosin heads will attach and pull the actin filaments toward the center of the sarcomere or toward the center of the H zone, what have you. Kind of like in this picture here. I like this picture. I don't usually show it during microanatomy because the colors are reversed, you'll notice. But I like this idea here is that the myosin heads are like these little guys playing tug of war with the actin filaments and pulling them inward toward the center of the H zone, or the center of the sarcomere, whichever you prefer. So that's how that works. And then the calcium signal does end. The 5th step, I think, is a very important one, is that the calcium signal has to end. It ends within 30 milliseconds as calcium is actively pumped back into the sarcoplasmic reticulum. So now what we're going to do is we're going to change gears and we're going to talk about energy and ATP. And we're going to kind of go back over some of these steps when we talk about the roles of ATP and EC coupling, and specifically what happens with myosin cross bridges. But to start talking about that, we have to briefly discuss ATP, because I don't always think it's entirely well understood at the level of general biology. I always like to discuss it very briefly in the context of anatomy, and specifically what we're interested in here. So we said at the beginning of the lecture, there are 2 things that cause the actin and myosin to begin moving to overlap each other. The calcium story was just discussed, and now what we're going to do is we're going to turn to the ATP, or energy story. Okay, so this is a molecule of ATP. We have adenosine at one end, and we have 3 negatively charged phosphate groups on the end. And what gives that 3rd phosphate group its incredible energy is the fact that it is negatively charged and it's being attached to something that already is very negatively charged. You know, both of these phosphates that are already here are negatively charged, and this 3rd phosphate just doesn't really want to stay. So it requires a molecule of it requires energy, usually in the, we talk about it being in the form of glucose, but any sort of nutrient can give energy into the system, enough energy that we can take this inorganic phosphate molecule and just very briefly and not with great stability attach that negatively charged phosphate to the end of the ADP. So we have our ATP, which is adenosine triphosphate, but it's created by taking a molecule of ADP, which is slightly more stable, adenosine diphosphate, 2 phosphates, forcing a molecule of phosphate onto it. So we have our phosphate group then attached to the ADP, creating ATP. And this, of course, is an endergonic reaction. When we take the phosphate and we stick it onto 2 other phosphates, it is an endergonic reaction. So what we're looking for then is very quickly, because this is such an unstable structure, Very quickly, we're going to take that ATP and make something happen with it through an exergonic reaction, allowing energy to leave the system. And this then allows the inorganic phosphate to be separated and to have your ADP as well. So let's take a look then at the cross-bridge cycle. So this basically points out there's a couple things going on. When you look at this picture, you have to understand that the calcium story stands. The calcium, you could actually see the calcium attaching to the troponin in this picture, in all four of these pictures. So we're going to talk about this cycle, but notice that the troponin has calcium bound to it at all times. So we're already assuming that the tropomyosin blockade has been removed, that the actin active zone is open, and that the myosin is free to interact with the actin because it's not blocked from doing so, right? But we're going to talk specifically about what happens with ATP here. So we're going to start with this picture. Let's see. We're going to start with the picture at the top here. It's called cross-bridge formation. What we have is that we have this cross-bridge where the myosin head has attached to the actin. And what you'll notice is that the myosin is actually in its cocked form at this point. So if you take your little sketch in your notebook and take a close look at it, notice that it is in its cocked form during this first stage. It's in its energized form. The next thing that's going to happen is that the myosin head is going to be relaxed. It's going to go from its cocked form to its relaxed form. And in so doing, it actually moves the actin filament. So the myosin head pivots and bends as it pulls on the actin filament, pulling it inward toward the middle of the H zone or the middle of the sarcomere. And interestingly, this stage is called the power stroke. Even though it's actually the relaxation from the energized state into the less energized state. It's called the power stroke because this is, in fact, the stage at which the actin moves. So this is the only place where actin actually moves, all right? Even though there's no energy expended at this point, very interestingly. Okay, so in between stage 2 and stage 3, look what happens. ATP comes into the system. ATP is going to attach to the myosin head. In so doing, it gives just enough of a conformational change to the myosin head that the myosin head is able to release from the actin active site. So third stage is just the detachment of the cross bridge as ATP attaches to the myosin head. Now, as the ATP then splits apart into ADP and inorganic phosphate here, that is, as we just stated, that is an exergonic reaction. And so that is enough to cock the myosin head, to bring it backwards so that it is in position to be ready for the next cycle. So as ATP is split into ADP and inorganic phosphate, the myosin head cocks. All right, so this is a cycle, so you could actually start at any point during this and say, you know, this is where it's all started. But here we go. Then with the myosin head is cocked, and then the first step again is that the cross bridge forms, where the myosin head is interacts directly with the actin active site. And you'll notice that the ADP and the inorganic phosphate are still there, attached to the myosin head. So now, when we go to the next stage, we say this is where the cross bridge happens. What's the next stage? Well, it's cocked at this stage. Remember, in the second stage, it goes from cocked to relaxed, and in so doing, it moves the actin filament, but this is not an energetic movement. In fact, this is a movement where you're going from an energetic state to a relaxed state. So the ADP and the inorganic phosphate fall off the head, ready to be recycled into our next molecule of ATP, by the way. And in so doing, the myosin head moves, pulls the actin filament inward. And this keeps on happening. It happens, you know, again, we mentioned earlier that for every action potential that comes down the myosin, down the motor neuron, and then comes along the sarcolemma, that it lasts about 30 milliseconds. Like, you know, the calcium influx lasts for about 30 milliseconds. So technically, this could keep happening for 30 milliseconds as long as you still have calcium binding to the troponin and that the actin active sites are exposed. You could keep on doing this as long as you have enough ATP, and technically you should always have enough ATP because inside of a muscle fiber, you remember you always have those glycosomes, and you have plenty of mitochondria, and you have the myoglobin, so you have the organelles to make lots of ATP, and you have a ready supply of glucose, and you have a ready supply of oxygen, and so you can keep on doing this for quite a long time as long as the calcium is present. So for every action potential that comes along, the calcium interacts with the troponin for about 30 milliseconds, and this stroke here can keep on going and keep on coming back and going through this cross bridge cycle for an entire 30 milliseconds. And so that is the story of calcium and the story of ATP and how they interact together to perform both EC coupling and cross bridge formation.